Hello everyone. Now in this video we are going to discuss regarding internal structures of dicotyledon leaf as well as monocotyledon leaf. First of all we will discuss regarding internal structure of dicotyledonous leaf. See, the transverse section of dicot leaf shows dorsiventral or bifacial structure, and the internal structure is differentiated into epidermis, that is upper and lower epidermis, central mesophyll tissue and vascular tissue. And here are some striking features of dicotyledonous leaf. First, it shows epidermis, mesophyll tissue and vascular bundle. Secondly, unicellular or multicellular trichomes are present on the epidermis. Third, the size of vascular bundle is larger than vascular bundles present in the lamina. Mesophyll is differentiated into palisade and spongy parenchyma. And lastly, guard cells of stomata are kidney shaped. So let's discuss in detail the internal structure of dicot leaf with the help of this diagrammatic representation of tiers of dicot leaf. Now look at the diagram here. This diagram shows first region, that is epidermis. Here this is the upper epidermis, this is the lower epidermis. As we discussed that the transverse section of dicot leaf shows dorsiventral or bifacial structure. Dorsiventral or bifacial, there are two sides, dorsal as well as ventral. That's why this tiers is known as, or this structure is known as a dorsiventral or bifacial structure. Now this bifacial structure shows first the outermost layer, that is epidermis. This look, the leaf is covered on both surfaces by a single layered epidermis, and the cuticle is present on both sides of the epidermis. Now the stomata are observed on both surfaces, but they are numerous on the lower epidermis. Look at here, in the epidermis, here in lower epidermis as well as in upper epidermis there is presence of stomata. The guard cell consists of chloroplast which help in closing and opening of stomata. So these are kidney shaped guard cells of stomata which helps to open and close the stomata. If stomata are present only on lower epidermis, then leaf is said to be hypostomatic. Whereas when stomata are present on upper epidermis, the leaf is known as epistomatic. Unicellular or multicellular trichomes are present. In epidermal tissue, we can see unicellular or multicellular trichomes. The epidermis checks excessive transpiration due to cuticle. This cuticle on the epidermis, it helps to reduce the transpiration or loss of water from the leaf. The stomata are helpful for exchange of gases between the plant and the atmosphere. Here below the stomata there is a one cavity called as a respiratory cavity, and this cavity stores whatever gases generated or water vapor generated in tissues, and when stomata is open there is a exchange of gases between plant and atmosphere. The transpiration also takes place through stomata pores which help in maintaining temperature of plant body. The epidermis also prevent entrance of pathogens into interior of the leaf. As the epidermis is outermost continuous layer, it prevents the entrance of pathogens or infectious organism, microorganisms inside the leaf. Another most important function of epidermis is the protection of soft internal tissue of the leaf from mechanical injury. So this is all about the first region of bifacial structure of leaf, that is epidermis. Now the second region which lies between two epidermis, that is called as a mesophyll tissue. This tissue from upper epidermis to the lower epidermis, this innermost inner part of the leaf is called as a mesophyll tissue. Now see, it is chlorophyllous tissue that lies between upper and lower epidermis and differentiated into two types of tissue, that is first palisade tissue and second that is spongy tissue, or we can say palisade parenchyma or spongy parenchyma. Now the palisade tissue are usually these are palisade tissue are usually elongated, compactly arranged in single or more columnar layers, and these are usually arranged below the upper epidermis, but sometimes it may be arranged below the lower epidermis. For example, in case of eucalyptus, the palisade tissue is present below the lower epidermis, where both the surfaces of leaf are equally illuminated. The leaves which receive more sunlight develop more compact palisade parenchyma in comparison to the leaves which develop in shady places. They are very useful in photosynthesis as they consist of chloroplast. Now a region below lower epidermis is occupied by spongy parenchyma. This is the spongy parenchyma. So usually they are composed of loosely arranged, irregular and thin-walled chlorophyllous cells, and due to presence of large air spaces they are helpful in exchange of gases between cells and the atmosphere. They are photosynthetic in function and they are also concerned with the storage of food material. So this is the mesophyll tissue divided into palisade and spongy and present in between the upper and lower epidermis. Now the third important region of this leaf is vascular bundles. This mesophyll tissue is characterized by presence of vascular bundle in between. These are vascular tissue which constitute the conducting tissue system in the midrib of leaf. The xylem always lies toward the upper surface and phloem towards the lower surface. Xylem conduct water, raw material and also gives mechanical support to the leaf. And phloem serves for translocation of prepared food material from mesophyll of leaf. The vascular bundle of leaf is surrounded by parenchymatous bundle sheath and it has direct contact with the conducting cells of the vascular bundle. In the midrib, below and above the vascular bundle, mechanical tissue like collenchyma, sclerenchyma or turgid parenchyma are present and it helps in giving mechanical strength to the leaf. For example, this is the tears of sunflower leaf which showing the anatomical details. So that's it regarding internal structure of dicotyledonous leaf, which shows the striking feature that it is a bifacial structure having upper and lower epidermis in which the mesophyll tissue is divided into palisade as well as spongy tissue. Next, we are going to discuss regarding internal structure of monocot leaf. Now, in transverse section, the monocot leaf shows isobilateral structure, that is monofacial structure. Monocot leaves are mostly erect and more or less both the surface usually receive direct and equal amount of light, and it is differentiated into epidermis, mesophyll and vascular bundles. See, upper epidermis consists of bulliform cells is the striking feature, and mesophyll is not differentiated into palisade and spongy parenchyma. Let's see the internal structure of monocot leaf. This is the TS of maize leaf showing the internal structure. Here we see the first region, that is the outermost region of the leaf is epidermis. That is it is the outermost protective layer found on both the upper and lower surface of leaf, and the epidermal layer are uniseriate, that is made up of a single layer of cells. The epidermal cells are more or less oval and compactly arranged. So this is the epidermal layer. This one is upper epidermis, this one is lower epidermis, and epidermis is covered with the cuticle. Now the outer wall of epidermis is cuticularized, and the upper epidermis consists of bulliform cells. This is the bulliform cells, and they are hygroscopic in nature and causes rolling of leaf lamina. Stomata are present on both the epidermis. Stomata are present on both upper as well as lower epidermis. Here in case of monocotyledon leaf, the stomatal guard cells are dumbbell shaped. Now the next part, that is mesophyll tissue, the tissue present in between upper and lower epidermis. It is called as a mesophyll tissue. As the monocot leaf is isobilateral, the mesophyll is not differentiated into palisade and spongy tissue. Here we can see a one type of cells. That's why we call it as a isobilateral. The spongy tissue are isodimetric and chlorophyllous cells. Large intercellular species are present among the cells, and mesophyll performs the function of gaseous exchange as well as photosynthesis. Now this is the internal structure is characterized by presence of vascular bundle. Here we can see the vascular bundle. These are the vascular bundles. These are nothing but the leaf veins, and in monocot leaf there are parallel veins are present, or parallel venation is present. Now let's discuss regarding vascular bundles. Here the vascular bundle are closed. Each bundle is surrounded by parenchymatous bundle sheath. Xylem lies towards the upper epidermis. This is the xylem which is towards the upper epidermis, and phloem lies toward the lower epidermis. This is the part of phloem which lies towards the lower epidermis. The sclerenchyma cells are found to be scattered in patches on both the ends of large vascular bundle. Here is the sclerenchymatous tissue. Look at here and here. Then there is a presence of sclerenchymatous tissue. Now this sclerenchymatous tissue provide mechanical support to the leaf lamina, and it conduct the food material, water and inorganic substances. Now this is regarding internal structure of monocot leaf. The striking feature of monocot leaf, that is presence of bulliform cells, this one into upper epidermis. This structure, that is mesophyll, is not divided into spongy and palisade tissue. So these are striking features regarding internal structure of monocot leaf. Here we done with our topic number 5, in which we have discussed regarding internal structures of monocot root, stem, leaf, as well as dicot root, stem and leaf. Thank you.